Sorry, David, I'm going to present in Spanish. Sorry. Ok. El doctor David Rose estudió su licenciatura y maestría en la Universidad de Birmingham, Young, y su doctorado en Ciencias de la Alimentación de la Universidad de Purdue. Actualmente es profesor en los departamentos de Ciencia y Tecnología de los Alimentos y Agronomía y Horticultura en la Universidad de Nebraska, Lincoln. El Dr. Ross es miembro fundador del Nebraska Food for Health Center, un centro de investigación que se enfoca en mejorar la salud humana al vincular la agricultura y la producción de alimentos con el bienestar y la prevención de enfermedades a través de la investigación del microbioma intestinal. La investigación del Dr. Rose se centra en mejorar el impacto de los cereales y las legumbres en la promoción de la salud. El trabajo aborda dos áreas: la modulación de la microbiota intestinal hacia una composición y función más saludable mediante el uso de cereales integrales procesados y legumbres o componentes de estos alimentos, y el desarrollo de herramientas y estrategias para aumentar el consumo de alimentos ricos en fibras elaborados a partir de cereales y legumbres. El objetivo de la primera área es determinar cómo se pueden usar los componentes dietéticos de granos y legumbres para modular el metabolismo microbiano en el intestino y promover la salud del huésped. El objetivo de la segunda área es aumentar la ingesta de fibra dietética por parte de los consumidores mediante la creación de entornos de elección en los que sea más probable que los consumidores elijan alimentos ricos en fibra dietética. La investigación del grupo del Dr. Rose y sus colaboradores se ha publicado en más de 100 publicaciones revisadas por pares que han recibido más de 3.500 citas. El reconocimiento a sus contribuciones en el área de la fibra dietética y los granos le ha permitido al Dr. Rose recibir el premio de Young Scientific Research Award of the Cereal Grains Association, actualmente American Association of Cereal Chemistry International, y el premio John Halverson Memorial Lectureship de la División de Molienda y Panificación de la misma asociación. El Dr. Rose actualmente se desempeña como un miembro del consejo editorial de 4 revistas en el área de investigación sobre agricultura, alimentos y microbioma. Es vicepresidente del proyecto multiestatal NC 213 del Departamento de Agricultura de los Estados Unidos y presidente del Comité de Graduados del Departamento de Ciencias de los Alimentos en su país. It's for all a pleasure that you present your conference. Welcome, Dr. Rose.
Thank you for that introduction and for the invitation. I'm happy to be here. I hope that I can be understood. I wish that I could give my presentation in Spanish. My wife speaks Spanish very well, but I don't know that she would be able to present my research to you very well. So anyway, thank you again for that invitation, for the invitation and for the introduction. So today I am going to tell you 2 stories. The first story is going to be focused on how grain processing can affect fiber fermentation by gut bacteria and how that might impact metabolic health. I'm going to tell you a related second story that is focused on common bean, this picture up here on the top. So, and I'm doing this because in preparing for this talk, I was looking up fiber intake in different countries, and I noticed that Mexico's in a much better place than United States as far as fiber intake, and it's primarily due to the legume intake that you have there as being kind of a staple food in Mexico as opposed to the United States. So thought I'd throw in a story about some work we've been doing really recently on common bean. Nebraska is a very high producer of beans, pinto, black beans, and great northern beans. Okay, but first to talk about grains and processing. So grains that are composed of kind of different cell wall polysaccharides than a lot of other foods that we eat. So on the top there, we have the primary cell wall on the top left, and then on the top right is the secondary cell wall of dicotyledonous plants. And so these would be like fruits and vegetables. When we talk about grains, though, they have a different cell wall structure. It's shown there in the bottom two panels. And in grains, as we eat them, this would be their secondary cell wall. So bottom left panel there is what whole grains and whole grain dietary fibers look like. So we have three primary polysaccharides there. We have cellulose, which would be in these microfibrils kind of going across the panel. And it's kind of connected by arabinoxylans or sometimes heteroxylans if they have many sugars in them. And these are cross-linking that cell wall together. And then we can also have beta-glucan, mixed linkage beta-glucans in some cereals in these cell walls. Not so much in wheat, but in other grains. The dietary fiber concentration in different grains varies quite a bit. So you see in the top panel there, rye having a lot of dietary fiber relative to the other grains, and down to the rice, brown rice, which would be the lowest. And you see that the majority of those fibers are cellulose and arabinoxylan, those two cell wall polysaccharides. And then there's smaller amounts of beta-glucans, and then some non-cell wall polysaccharides, like the fructans and so forth. Dietary fibers differ within a plant type. So this histogram here on the bottom is just showing the variation in dietary fiber concentration and these are some European wheats, but the point is that it varies quite a bit among different types of wheat or other grains. So the story that I'm going to focus on today focuses on wheat and brown rice. So you can see where they fall relative to the other grains in their fiber concentration. So when we eat fiber, what does it do? It has a lot of health benefits, and many of those health benefits are related to how it interacts with bacteria in the large intestine. So on the left-hand panel there, I have a picture that shows what the colon looks like when somebody's eating a fiber-rich diet versus a fiber-free diet. And the main difference that you see here is the size of that mucus layer. So fiber helps to maintain that mucus layer and to maintain the integrity of the intestinal barrier. And when fiber is lacking, microbes sometimes can start to metabolize the mucin layer and start to penetrate the cells that line the large intestine, and some negative things can happen there. So we need to make sure to have enough dietary fiber in our diet to feed those gut bacteria and maintain our cell integrity, and also to provide some health benefits. Most of the health benefits related to fiber have been attributed to fermentation of dietary fiber. And when gut bacteria ferment that fiber, they can produce different short-chain fatty acids, which are byproducts of their metabolism. And those short-chain fatty acids are absorbed by us and influence all sorts of pathways related to metabolism and also cardiovascular health, also the brain, lots of different pathways. The plot there on the right mentions butyrate as one specific short-chain fatty acid that's produced that has shown, I would say, the most health benefits. And there are other short-chain fatty acids that are also produced that can have health benefits, but may not have health benefits, as we'll see in a minute. So, really important to maintain a high fiber diet to feed those bacteria, and to have that fiber able to be fermented by gut bacteria. So unfortunately with grains, not a lot of that fiber is accessible to gut bacteria. So if you look at the panel on the left, there's a picture of wheat bran when it's recovered from bread. And you can see the particles of wheat bran there, together with some starch granules and so forth. A little bit of structure there on the particles. And then the right-hand panel there showing wheat bran recovered from feces. You still see that there's a lot of structure there. So not a lot of those dietary fibers have been able to be fermented by gut bacteria. And in the bar chart on the right, you see that. So this is a human feeding study where they fed subjects either wheat bran or some fibers from apple or cabbage or carrot. And you see in comparison with fruit and vegetable fibers, wheat fiber is poorly accessible to gut bacteria. So only about 1/3 of the dietary fibers are able to be fermented by the gut bacteria, and about 2/3 pass through. And this 2/3 isn't contributing to those beneficial short-chain fatty acid production and some other things that are needed to access those benefits of fiber. So this kind of represents an opportunity for us to do research into how can we increase the amount of fiber that's accessible by gut bacteria, and therefore increase the potential health benefits that are provided by fiber. So in this plot, we see some micrographs that show the cellulose and the arabinoxylan structure in a porridge or an extruded product. So in the porridge on the left, you see that there's quite a bit of structure still left there in the porridge. So after grinding this product into a flour, boiling it with some water, and then looking at it, it looks like there's still quite a bit of structure intact. In contrast, on the right, when we extrude this product, which subjects this flour to very high temperature and shear and pressure, breaks up that structure, and potentially allows bacteria maybe to ferment more of this carbohydrate. So this was our hypothesis, is that if we take grain and extrude it, breaking up those structures and so forth, that the bacteria will be better able to access those fibers and break them down, and then produce more of those beneficial short-chain fatty acids, contribute to health benefits. So this slide kind of encapsulates the main activities that we do in my lab. So for this study, it shows the experimental design. So first of all, we hypothesized that grain processing, especially severe processing like extrusion, would enable more of those carbohydrates to be fermented by gut bacteria. So we looked at 5 processing methods here. We looked at the 2 that I showed you the picture of, porridge. That's kind of a minimal processing, and extrusion at the bottom, a very severe process. And then we also looked at 3 forms of baking, which were an unleavened bread, and then yeast leavened bread, and then a sourdough fermented bread. And, these, at least the unleavened bread and the yeast leavened bread, these don't really affect the cell wall polysaccharides, but potentially could be affected in the sourdough bread. So we have these 5 products, all processed from wheat, and we subjected them to digestion in a test tube. And this was to get rid of all of the digestible components, because normally if we were to consume these products, they would be digested before gut bacteria see them. So we have to subject all of these to digestion using different digestive enzymes and buffers to break down starch and protein, fats, and so forth. And then we subject that to dialysis so we can remove those digested components, leaving behind the undigested components, which represents what the gut microbiome would normally see. Then we take stool samples that we collect from subjects, and this is our source of gut bacteria, and we mix that in some media with our digested processed grains. And we allow that to ferment under anaerobic conditions for a period of time, and then we look at changes in microbiota composition, and we also look at carbohydrate degradation and short-chain fatty acid production. So we found some unexpected things here. So if you look at the panel on the top left, this was our primary outcome. Primary outcome was carbohydrates utilized, or the percent of carbohydrates that were fermented. And remember, we wanted to increase this because we thought that would be beneficial, since benefits of fiber are related to fermentation. So what we saw was that the carbohydrates that were fermented varied a lot based on the different microbiome that we used. But they separated into 2 groups. They separated into a group that could not ferment very much of those carbohydrates, around 20%, and then another group that could ferment about 10% more of those carbohydrates. So we classified these into 2 groups called high and low carbohydrate utilizing microbiomes. And when we looked at the effect of processing in these 2 groups, we saw something very interesting. In the bottom left panel, we saw that in the high carbohydrate utilizing group, that they had a response to processing. So, and it's exactly what we expected, which was that extrusion enabled more of the carbohydrates to be fermented than some of those less severe processing methods. But when we look at the low carbohydrate utilizing microbiomes, we don't see any differences between the samples, between those processed samples. So we say that these low carbohydrate utilizing microbiomes didn't respond to the effective process. We saw the same thing with butyrate production. So we see differences in the high carbohydrate utilizing group and no differences in the low carbohydrate utilizing group. What we did not expect was not to see low butyrate production from the extruded samples. So we expected that when we extruded a product that we would get more carbohydrate fermentation, which is what we got, but we thought that we would also get more butyrate production, which we didn't get. So we looked at the changes in microbiota composition, and if you look at the heat map on the top right there, those are the changes in different bacteria during fermentation that varied across the different processing methods for the high carbohydrate utilizing group. And what we saw was that extrusion caused a decrease, you see that in blue here, a decrease in some of those beneficial bacteria that are involved in cell wall polysaccharide metabolism. And instead you saw an increase in these microbes, Enterococcus, Staphylococcaceae. These are microbes that grow quickly and that grow on sugars. And so this was very unexpected. It explains why we didn't see this increase in butyrate production that we expected. And I'll just mention here, this bottom heat map is the changes that occurred in the low carbohydrate utilizing group. And we see fewer changes, but similar changes. So what we proposed was happening is that when we take the flour and we extrude it, so we'll follow this bottom pathway here, when we extrude that flour, we break up that cell wall extensively. And so that when it comes in contact with the microbiota, those specialized cell wall degrading bacteria that are beneficial and produce butyrate, they are not able to outcompete fast-growing bacteria that can use those sugars quickly. And so the butyrate producers decrease, and those fast-growing non-butyrogenic carbohydrate utilizing bacteria take over. And in contrast, when we treat something with a different processing method, here like sourdough, or it could be like boiling into porridge, which we're going to talk about next, those cell walls largely remain intact. And so those fast-growing bacteria cannot access those carbohydrates, but those specialized bacteria that produce butyrate are able to access it and produce that butyrate. So unexpected results here, but still quite interesting. So we took this further. We wanted to see if these results would translate into an in vivo model. So we took two microbiomes from our previous study. Those two here in the top left panel show we took actually microbiome 1 and 2, which were in the two different groups. they represented the mean of their group. So they're not the extremes, but instead the mean of their group. And we transferred those microbiomes into germ-free mice to make human microbiota associated mice. So now these mice have a human microbiome in them, and they have one that has high carbohydrate utilizing capacity and one that has low carbohydrate utilizing capacity. We randomized these mice into 5 groups. They were consuming either a low-fat diet, which was a control, a Western diet, which was another type of control that had 45% fat and a bunch of sucrose in it. And then we also had our processed grains added in with that Western diet. So we had a porridge and an extruded diet. And then we also included brown rice here so we could compare the effect of processing to the effect of grain type. So if we look at the two brown colors, the yellow and brown. Hello? Does someone have a question?
No. Mónica, por favor, ¿puedes apagar tu micrófono? Gracias. Sorry, Dave.
That's okay. I just don't want to skip someone if they've got a question. Okay, so with these 3 diets, we can look at the effect of processing by examining differences between porridge and extrusion, and we can look at the differences between grain type by looking at the whole wheat diets versus the brown rice diet. We chose brown rice because it's so different from wheat as far as it has very low fiber concentration. When we look at the composition of these high and low carbohydrate utilizing microbiomes, we see some important differences. First of all, if we look at the diet of the person that those 2 microbiomes came from, we see that the high carbohydrate utilizing microbiome came from somebody that had a very high fiber intake and especially a very high whole grain intake. So this is good. This is kind of expected that perhaps the microbiome had been acclimatized or used to metabolizing those carbohydrates because that person ate so much whole grain. And the low carbohydrate utilizing microbiome was more similar to what we would expect, a fiber intake kind of moderate and a whole grain intake fairly typical. In panel B there on the right, we see 2 measures of diversity. We see observed ASVs, so this is like the number of different species that are there. We see that there's more different ASVs in the high carbohydrate utilizing microbiome, which we would expect. When we look at Shannon diversity, we see no difference. Shannon diversity measures not only the occurrence of different bacteria, but also it takes into account their abundance. So if we have a lot of microbes that are present in very low abundance, then Shannon diversity will decrease. And so that's why we see that those 2 bars are about the same. When we look at the composition in this heat tree here, we see very different composition. We see that the high carbohydrate utilizing microbiome differs in quite a bit in the abundance of the 2 major families of firmicutes, which are Ruminococcaceae and Lachnosporiceae. So the composition is quite different there, with the high carbohydrate utilizing microbiome having a higher abundance of those cell wall degrading genera. And then also we see that on the Bacteroidetes side, that the high carbohydrate utilizing microbiome has a lot more Prevotella which is associated with high fiber intakes. So we see these expected differences between these two microbiomes. So when we look at what happened in the mice during feeding, we see, so I have all these panels set up the same, where we have high and low carbohydrate utilizing microbiome separate, and then we look at all of the diets together within a microbiome. So in panel A, we have body weight, changes in body weight, and then panel D, we have body weight gain, and then we have body fat and lean mass in the other panels. So what I want to highlight, I've circled here in either red or green boxes. So the red box means that we have differences between the whole grain diets for both microbiomes. So if we look at the one red box here, we see that there's some separation between these different diets among the mice. So here, the extruded diets caused significantly more weight gain than the control Western diet, and also the porridge diet, shown there in yellow. We see some differences similar with the low carbohydrate utilizing microbiome, where the extruded sample was worse or caused the mice to gain more weight than the porridge sample, but it wasn't different from the control Western diet. In the green boxes, we see differences that only occur in the high carbohydrate utilizing microbiome. So for example, in panel B, we have differences between the extruded samples and the porridge sample. And in the low carbohydrate utilizing microbiome, we have none of those differences. And that's repeated across the rest of these panels. You see significantly lower body weight gain in the porridge sample compared with extruded sample in the high carbohydrate utilizing group, and not in the low carbohydrate utilizing group. If we look at blood glucose response, we see something similar. If we look at the glycemia over time after receiving glucose injection, extrusion was worse than porridge for both microbiomes. If we look at the green boxes, extrusion was worse than porridge in the high carbohydrate utilizing group, and in the low carbohydrate utilizing group, there were no differences. If you look at panel L, the bottom left or the bottom right, we clustered the responses based on all of the things that we measured in the mice. And what we see is that, first of all, on the x-axis, all 5 diets cluster exactly the same in the high and low carbohydrate utilizing groups. But the length of the branches are different between the two groups. So the branches are longer in the high carbohydrate utilizing group, which is consistent with all the panels that we see where there are more significant differences between the different diets, especially between the whole grain diets in the high carbohydrate utilizing group. And in the low carbohydrate utilizing group, those branches are shorter. And we see that reflected in the data where there are fewer significant differences between those diets. So now we look at changes in the microbiome. So these are 2 panels that are showing the beta diversity. Beta diversity shows or measures differences between samples. And I'm using two metrics here. We're using unweighted and weighted unifrac. So unifrac takes into account, unweighted unifrac takes into account the phylogeny of the different bacteria that are present and their presence or absence, whether they're there or not. And you see in panel A that the main differences between the bacteria in these mice is their carbohydrate utilizing group, or their fecal donor. So if they received the high carbohydrate utilizing microbiome, it's very different from the low carbohydrate utilizing microbiome, even though all of these mice were consuming, the same diets across those. We also see that the microbiome changes during the study from week 0 to week 11. If we look at the weighted unifrac, so weighted unifrac takes into account everything that unweighted does, but also takes into account the abundance of the different bacteria that are present. And what we saw in the weighted unifrac was very interesting, which is that there's a lot of spread between those different points for the high carbohydrate utilizing group and not for the low carbohydrate utilizing group. That's the same thing we saw in the mice data, where we saw that there was more spread among the different diets in the high carbohydrate utilizing mice than the low carbohydrate utilizing mice. We actually measured the spread of those points, and we see at week 11, the most important week, that there aren't differences in the spread of those points for the unweighted unifrac distance, but there are for weighted. So this tells us that the differences between those different diets that we saw in the mice were probably related to changes in the abundance of microbes, rather than whether they were just there or not. So we wanted to look at changes in the abundance of these bacteria over time. So we did that, and it's plotted here. So we have heat maps showing the log 2-fold change of the bacteria during the study. So if it's red, that microbe went up during the study, and if it's blue, it went down. And if it's gray, it means that bacterium was not present in that sample. And so we clustered the bacteria by phylogeny, and you see across the two microbiome groups, high and low, that the changes that occur are fairly similar. So if we look up there at the top at that cluster of firmicutes, but Lactospiraceae, that there's some kind of red colors that you see there for the Western diets, and more blue colors for the low-fat diet. And you see that for both high and low carbohydrate utilizing groups. Even though it's different taxa that are changing, they're related. And you see that down the whole heat map. You look in the middle at the Bacteroides, you see a lot of blue there, so they're decreasing in both the high and the low groups. So the changes are fairly consistent, but if we look at how the diets group, or diets cluster, so we look at the clustering of the columns in those heat maps, we see that the two extruded diets, the whole wheat and the brown rice extruded diets, they clustered together. And they're clustered separate from the rest of the diets. In contrast, in the low carbohydrate utilizing group, they cluster by grain type instead. So the whole wheat samples cluster together and the brown rice clusters separately. if we remind ourselves how the mice data clustered, in the mice data we saw that the extruded samples clustered together, just like we see in the high carbohydrate utilizing group for changes in the microbiome. And we don't see that in the low carbohydrate utilizing group. So this tells us that the changes that we see in the mice, the metabolic changes that we saw in the mice, were at least partially related to changes in the microbiome for the low but not the high carbohydrate utilizing group. So because the extruded samples were giving a similar response in the mice and also in the microbiome, we grouped those two together to see how they were different from when we were giving the mice the porridge. And so the plots on the left there, panel A and B, these show the bacteria that were related to extrusion, there in that olive green color, or porridge in the yellow color. So the bacteria in the high carbohydrate utilizing group were mostly members of Rruminococcacea and Lactospiraceae, all except for one. And in the low carbohydrate utilizing group, we saw that as well. It doesn't really make sense to cluster those two extruded diets in the low group. We just did it for comparison. We took those microbes and we did correlations between their abundances and some of the metabolic parameters that we measured in the mice, fasting glucose, weight gain, and so forth. And what we saw in the high carbohydrate utilizing group, we saw that those bacteria that were related to extrusion had a lot of highly significant correlations with those negative outcomes in the mice. So high abundance of these taxa was related to high fasting glucose, high percent fat, high weight gain, and so forth. We also saw that the taxa clustered exactly according to the treatment group. So in panel C, you see all of those green, they're all members of, or they're all associated with extrusion, and they're all up there at the top together. And then those taxa that were related to porridge are down at the bottom and have the opposite cluster, the correlations are in the opposite direction. But we don't see that in the low group. We see that the clustering is not by treatment group, and also the correlations are not as strong. We wondered what these bacteria were doing that could have been so detrimental. Because these bacteria on their own don't necessarily point to being negative. So we used a program that can predict what the function of those bacteria are, and then we plotted those differences between the extruded samples and the porridge sample. And we saw that for the extruded sample, we see very high pathways involved in sugar degradation and short-chain fatty acid production. This is kind of what we saw in vitro, where we saw that the mice, or the bacteria that were grown on the extruded sample had higher sugar degradation. And so, because these two pathways were lighting up so much between the extruded and the porridge samples, we looked at short-chain fatty acids in the cecum and the colon of the mice, and we saw increased acetate production in the colon of the high carbohydrate utilizing mice that were consuming extruded diets compared with porridge diets. So acetate is usually thought of as beneficial. It's produced from fermentation of carbohydrates and other components by the microbiome. And it can be absorbed and influence a lot of pathways in our body related to satiety and so forth. But it can also be used for energy recovery. So it can enter the tricarboxylic acid cycle or the citric acid cycle and contribute to energy production in the mice or in us. And so what we think is happening is that those bacteria that were associated with extrusion were fermenting more carbohydrates and producing this excess acetate that went to excess energy recovery. We did see different microbes that were associated with extrusion in vitro versus in vivo. So at the very beginning of the presentation, I showed you that extrusion was associated with Enterococcus and Staphylococcaceae. But in the mice, we showed that they were associated with different members of Ruminococcaceae and Lactospiraceae. Different bacteria, but we hypothesized that they were performing similar functions in vitro and in vivo. And in the panel on the left, we see that. So that panel shows the correlation between all the pathways and their abundances in vitro on the x-axis and in vivo on the y-axis. So these are they're different bacteria, but performing similar functions in the two systems. And we also looked at the function of those bacteria in vitro, and we saw the same thing we saw in vivo, which was that extrusion caused this runaway sugar degradation and short-chain fatty acid production. The take-home messages from this study were that in both microbiomes, the impact of processing was greater than the impact of grain type when we look at the outcomes in the mice, and that severe extrusion resulted in negative host health. I should emphasize the word severe in that bullet point. We used very severe extrusion because we wanted to maximize the effects of processing, and so other extrusion conditions that are more typical may not give the same negative response that we saw here. In only the high carbohydrate utilizing mice did we see that the effects of processing were related to changes in the microbiome, and that the grain processing affected the composition of the two major families of bacteria in the gut, which are Lactospiraceae and Ruminococcaceae. And that severe extrusion led to excessive acetate production from these families of bacteria that contributed to excess energy recovery. And then finally, when we look at the results in vitro, they were able to predict what would happen in vivo. That's very important for us. Okay, now I'll go on to the second story. Oh my goodness, do I only have 7 minutes? Okay, we'll go quick on this one. So beans have a lot of components that are likely to impact the microbiome. And, not just fiber, but other things as well, like polyphenolic compounds and so forth. And the literature is very sparse on exactly how beans can influence the microbiome. So we did this giant project where we took 299 genotypes from common bean and went through that in vitro fermentation that I talked about previously with 3 microbiomes. And you see down there in the middle panel how many samples this was. So a lot of samples. And then we used the genotype data to map back to changes in the microbiome. So we're trying to look at what genes in the genome of beans are related to changes in different gut bacteria. And we were not able to do this using our normal techniques, so we had to develop a more high throughput method for doing this these in vitro fermentation tests. So these are some pictures from that. We have automated a lot of the steps for sample preparation and for sample weighing and for in vitro digestion and fermentation to enable us to analyze this many samples and complete this study. So we started off looking at about 400 different features of the microbiome and how they related to the genotype data of these different beans. We only considered traits that had a significant amount of variance explained by the genotype. And so once we did that, we ended up with 214 different microbiome features that we mapped to the genome of beans. So we used GWAS to relate the changes in the microbiome to the genome of beans. And we had 214 features for all 3 microbiomes. So we have these Manhattan plots showing differences between the microbiome, and we had 214 of these for each microbiome. So we were not able to kind of interpret these results. And so what we did is we took all of these dots up here above this dashed line, which are significant, and we just plotted them on a histogram. And what we see is that there's a few hot spots in the genome that pop out. And we call them major effect loci. And I will focus on that highest dot there, MELC. So I'm going to skip to this here. MELC, we see, was significantly associated with a very particular region in the genome. You see there in the heat map. And if we look at the genes, or the genes that are involved, or the pathways that are involved in that area of the genome, it maps to an enzyme that is involved in biosynthesis of these compounds here on the right, which are saponins. And so what we think is happening is that saponins, which have these sugars attached to them, that these Lactospiraceae and Ruminococcaceae that are beneficial bacteria have enzymes that can remove these sugars and metabolize them and produce butyrate, which contributes to some health benefits. And the aglycone, the saponogen, can also potentially provide some health benefits. May also be antimicrobial for some of the negative bacteria in the gut. If we look at one specific feature, which is Faecalibacterium, which is a very beneficial bacterium, and we look at the allele frequency in our different beans, we can see that there is variation within a market class for these beans. So, according to this, pink beans have a large difference within market class, and then across the market classes, they give the strongest response to this MEL, which is beneficial, and with those that have the minor allele here. And if you look at this picture on the right, if you were to grab 10 random pink beans from that panel, you'd have half of them having the minor allele and half of them having the major allele. And interestingly, we saw with Great Northern beans, there were no beans that had the minor allele representing this positive effect of Faecalibacterium. So, in summary, I'm sorry I went through this one fast. In summary, our automated microbiome screen platform can be used to screen a large diversity of crop plants to identify their effects on the microbiome. And then we can use those microbiome features in GWAS to identify regions of the plant genome that influence the gut microbiome. And microbiome active traits can be used in the development of crop breeding programs focused on human health. And then finally, variation, a lot of variation exists across market classes, but also importantly within market classes of beans. All right. That's the end of my presentation. Thank you for your attention, and if we have time for any questions, I'm happy to take them.
Okay. It's nice your work. Thank you very much. And now we are going to begin the questions. Por favor, si alguien quiere usar el micrófono, podemos usar el micrófono. Si alguien necesita traducción, agradezco mucho a Ariadna, que nos va a hacer favor de apoyarnos, la doctora Ariadna. Y de igual forma, ustedes pueden escribir sus preguntas a través del chat. ¿Ok? We are waiting. I have a question, David. This area, not is my specialty, but I have the question: what is the methodology that you use to know the bacterial microbiome?
Yeah, we use 16S Rrna sequencing, which is very common these days. So basically, we isolate this the DNA from the 16S region of the bacterial genome. And this is a region of the genome that is conserved across bacteria, but also has some variable regions that allow us to separate into the different types of bacteria, but just to pick up bacteria. Okay. So it's not a classical microbiology. It's related to DNA and so forth.
Okay, thank you. Esperamos.
Tengo una pregunta, doctor Mario.
Sí, Vanessa.
¿Me escuchan bien? Sí. Thank you so much, Dr. David. It was an excellent presentation. I have two questions for you about the first part of your presentation. The first one is, do you use dietary fiber or indigestible fraction in the in vitro fermentation of the wheat? And the second one is, what is the factor to which you attribute the separation of the two groups in high and low fermentation?
Okay, thank you. Those are good questions. So the first one, we used the indigestible fraction. So we did not use only the fiber. So it was not 100% fiber. It was mostly fiber, but it also had some other components in it. And then as far as separating into those two groups, I am not sure what separates those into the two groups, but based on the dietary data that we got from the two subjects that we did select for the mouse study, I suspect that diet is playing a big role in separating into either a high or a low carbohydrate utilizing group, where probably all of those subjects that were in that high carbohydrate utilizing group were consuming either more fiber or more whole grain specifically than those that were in the low carbohydrate utilizing group.
Thank you. Can I ask you one question more?
Yeah.
Yes.
About if the AIM platform is free?
So the AIMS platform is a custom thing that we have built in our lab, and so if we have opportunity to collaborate with people, we can use it, but we don't have any kind of setup where it's like a fee for service kind of thing. So yeah, it's and it's not plug and play either. There's a lot of steps that are automated, but it takes a lot of experience and so forth to be able to run through that. So.
Okay, thank you so much.
Yeah, we have done quite a few. I show you the beans here. We've done also several different types of grain through that. And I showed you the pathway that lit up for saponins because it was the largest peak there. Those other major effect loci that came up are related to other components in the bean. So as you would imagine, some of them are important in like pectin biosynthesis or starch biosynthesis. There's one of them that is really mapping to the flavonoid biosynthesis pathways. So it's enabled us to look at these different beans and identify a lot of pathways that are influencing these gut bacteria.
Another question, please.
Looks like there's a question in the chat. It says, according to your knowledge, the production of short-chain fatty acids by the fermentation of starch polyphenol complexes is good? Yeah, I mean, according to my knowledge, I would say yes. We haven't looked at those specifically in my research, starch polyphenol complexes. We have looked at resistant starch, but it wasn't generated as a result of interaction with polyphenols. But that fermentation of starch, resistant starch, is generally positive.
Okay. Thank you, Hector. Thank you, Dave. Another question?
Yo tengo una pregunta.
Sí, adelante.
Okay, thank you for your excellent presentation. I have one question. Which beans do you think are the best for the diet and why?
I think all beans are great, but I mean, if we look at this box plot here, for this specific outcome, the increase in these beneficial Rrna and Lactobacillus, especially Faecalibacterium in beans, looks like the pink beans are the best. And then the white beans are the worst, with Great Northern being the worst. These navy beans are also white, but at least we see some that have a minor allele. So, and they're different between within the market class. So it doesn't really separate perfect by color, right? Because we have pink being the highest, and then we have pinto, which is also pink, and small red, which is more or less pink, and then black, which I would think has even more color, but it comes in the middle. So anyway, with this specific outcome, pink's the best, but it doesn't come out like that when we look at other MELs.
Thank you.
Mm-hmm.
Gracias, Carolina. ¿Alguna pregunta más? More questions?
I have a question.
Adelante, Ariadna.
Congratulations, Dr. David, for your amazing work. It was a very nice presentation, and I can say that I learned a lot. This is a new topic for me. And I have a doubt. Do we have any knowledge regarding the cell wall degrading enzymes the bacteria in which you saw changes have that could explain your results?
Yeah, that's a good question. We did not measure any enzyme activities in these studies. So I don't have that information. It's well known that these bacteria that are significantly different and significantly associated with these different treatment groups are involved and at least have genes involved in carbohydrate degradation. And that's really why we see, when we look at their function, that sugar degradation is so high. I'll just go back to this one here, where sugar degradation is super high, because these microbes have those pathways abundant in their genome. We also see, I guess, fifth down on the list, polysaccharide degradation, very important as well. And we see those same differences in the heat map, just not as strong as sugar degradation. And they're taking these sugars and converting them to short chain fatty acids. So,
okay.
Yeah, looking at the enzyme activities would be interesting. We did not do that here.
Okay. Are you planning to do it, or that's an interest for another group?
We've talked about doing it in other studies. We didn't have any plans to do it here specifically, but it's a good idea.
Okay. And I have another question. Regarding the legume seed, the bean seed, is the cell wall of a bean similar to the cell wall of the wheat? Or
no, they're quite different.
Yeah.
Quite different. So beans are a dicot, so they're going to be, sorry about that. They're going to be the top there. They're actually more like the top left. So they're very high in cellulose, pectin, xyloglucan, those polymers instead. And they're much more fermentable. They're going to look more like the apple, cabbage, and carrot than like the wheat bran.
Okay, so we could expect different changes in the microbiome.
Yes.
Yeah.
Yes.
Compared with the wheat effect.
Yeah.
Okay. Thank you very much for your kind presentation and your responses.
Thank you. You're welcome.
Gracias, Ariadna. ¿Alguna pregunta más? Yo tengo una pregunta, la escribí en el chat. I have a question, I write in the chat. It is possible that the bacteria that you have identified as beneficial is possible use it as probiotic?
So the bacteria that we identified here are not on the market or sold as probiotics right now. It doesn't mean that they could never. If we went after specific taxa that had certain characteristics, then definitely we would want to market those or promote those as probiotic bacteria. The ones that we're identifying in this study, either study that I showed really, are not available as probiotics. The last one here, this Faecalibacterium, this is one that could be a probiotic if anybody could grow it really well on a large scale. It's very difficult to culture, but very, very beneficial bacterium. And there's quite a few that are being identified that are potential as probiotics. We don't do a lot of that, but right now I will say we are looking at microbes that can reduce flatulence by the microbiome. And so those could potentially be probiotics that would have that property of reducing bloating and so forth from consuming dietary fibers. So these could potentially, but none of these that we identified are sold right now as probiotics that people can take.
Thank you, David. Not more questions? Okay, no. Okay, thank you very much. And if I can use to compare here the slide.
Yes, you can use the slides. You can
do
whatever you want. Yeah.
Okay. Thank you, David. I'm going to compare the constants.
Dr. David, I think that.
Okay. I'm sorry. Is here. Okay. I'm going to read the certificate that we will send. Instituto Politécnico Nacional, Centro de Desarrollo de Productos Bióticos. Award the present certificate to Dr. David Rose, Department of Food Science and Technology and Agronomy and Horticulture, Nebraska Food for Health Center, for his presentation as a speaker with the lecture entitled Grain Processing Can Affect Fiber Fermentation by Good Bacteria with Implication for Metabolic Health. Present virtually at the seminar of the doctoral program in development of biotic products, Yautepec, Morelos, 28th of March. Thank you very much.
Thank you. That's very nice. And thank you, everybody, for your excellent questions and your attention.
Thank you. Muchas gracias a todos por su asistencia. Solamente me restaría indicarles que en el chat está la liga para el registro, para los que no hayan registrado su asistencia. Okay. Thank you again and see you.
Thank you. Bye, everybody.
Que tengan buen día. Hasta luego.